drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Wheeler, thank you for joining me.
Yeah, thanks so much, Nick.
Do you want to just start off by introducing yourself a little bit and talking about what you do and what you've been studying?
Yeah, so I'm an assistant professor of neurology at Brigham and Women's Hospital and Harvard Medical School. And broadly speaking, my lab's interested in neuroimmune interactions that regulate behavior. So both organic behavior, typical behavior, but also when behavior is dysregulated, such as in neuropsychiatric disorders. And we're very much interested in this whole continuum of neuroimmune interactions across things like mood disorders, as well as addiction, and also more unconventional neuroimmune interactions like inflammatory diseases where there might be cognitive deficits. So that's really what we study in a nutshell.
Mm-hmm. And so, at a very, very high level, like when you are learning about biology at like the high school or college level, maybe you don't tend to talk about the nervous system and the immune system in the same class or in the same session. In fact, one of the things you learn about the nervous system is it's got the so-called privileged state with respect to the immune system and circulation. The blood-brain barrier is supposed to be there to keep things out of the nervous system that would ordinarily get into other tissues. And so you're not supposed to get a lot of infiltration of bacteria and bugs into the brain. You're not supposed to get a lot of infiltration of immune cells that are probably patrolling the periphery into the brain. Can you give us just sort of a basic sense of the major ways that the immune system and the nervous system interact and the extent to which the the immune system is in the nervous system?
Yeah, great questions, right? So I think, about 10 years ago or so, we really started to reassess this concept of immune privilege in the brain, as you mentioned it. And I think that was spurred partly by this discovery from Yoni Kipnis's lab about meningeal lymphatics. And basically, these are structures that can receive molecules from the periphery and then convey those signals, these are adjoining the brain, and then they can convey these signals back into the body. And that, together with a lot of other work in the field of neuroimmunology, has shown that probably the brain's not as privileged from peripheral stimuli as we think. And I'll give like a couple examples. So on the one hand, we know that if we deliver peripheral molecules just in homeostasis in wild type animals, let's say, they can actually penetrate past the BBB. And these are proteins like cytokines. They can penetrate past the BBB into the brain parenchyma, something like cortex, something like that. So not very far. But then if you start thinking about how immune privilege shifts in the context of disease, and even something that's not an inflammatory disease like psychological stress, chronic stress, there's actually a loss of integrity of this barrier that it becomes even leakier. And I think kind of the classic report of this was from Scott Russo's lab at Mount Sinai. So basically, endothelial cell tight junctions get a little bit looser. And so now peripheral stimuli can get into the brain. And so these are kind of like the basic mechanisms in homeostasis and this model of stress, or a few models of stress, where this is changed. And then in terms of the sites that can actually communicate with the brain from the periphery, I think there's several key ones to highlight. Not all of them are relevant to this paper. But first of all, there's the meninges, which is this kind of layer of tissues that is beneath the skull, above the brain, and it can receive and convey information from the periphery and also convey that to the brain. We also know that there are channels that can convey signals from the meninges directly to the brain, which is kind of this interesting idea that actual larger products can get into the brain in homeostasis. And then there's kind of unexpected sources of immune cells, like something like the skull bone marrow, where immune cells can be generated from the skull and also communicate with the brain. And so this is kind of like the broad, let's say, landscape of how, we think of sort of neuroimmune interactions in the
Hmm.
brain.
Hmm. So what I'm hearing is the brain and the central nervous system are protected. There is a blood-brain barrier. It does regulate what goes in and out, but it's not perfect. It's not keeping everything out. Some things can get in much of the time. Some things can't get in much of the time. Some things can probably get in if they have the right password or whatever. But also under pathological conditions, that barrier can start to break down, and things that shouldn't be getting in a healthy state can then start to get in.
Yeah, that's exactly right. And so, what we need to start to understand, both in the context of the healthy brain, but also perhaps more importantly for the diseased brain, is what is the logic between, what signals can get in and what parts of the brain do they communicate with? And in particular, what cell types in those regions do they actually talk to? And so, we need to understand, we need to have something akin to like a map or a wiring diagram of how these signals from the periphery and the brain crosstalk.
Mm-hmm, mm-hmm. Can you talk a little bit more about the meninges and how it works under normal conditions? I don't think most people know very much about this, except that people probably have heard of meningitis.
Yeah.
So what is the meninges doing, and what do we really know about it?
Yeah, I mean, so the meninges is this layer of tissues. I mean, it's broadly kind of composed of several different tissues. There's kind of anatomical positioning differences that control what cell types are where in terms of what immune cell types. But there's epithelial cells, there's immune cells that come from circulation that go into the meninges, and in some cases they can stay there for longer periods of time. In something like meningitis, you have this increased inflammation of the meninges proper. And so the meninges can, convey bona fide immune responses. And there are actual, let's say, immune structures that we know happen in peripheral tissues that can also occur in the meninges, like a bona fide immune cell differentiation. And I think more broadly related to neuroimmunology, what we're learning about the meninges is that the cells that are present there are actively regulating processes in the brain. And so I think, a recent paper that I read from Diane Mathis's lab, who's here in the immunology department, showed that if you ablate specific populations of immune cells, you can actually induce infiltration of those cells into something like the hippocampus. And it's consequential for behavior. And so, this continuum of what is going on in homeostasis versus something like meningitis, where there's inflammation, there's tons of immune cells, and there's dysfunction. There's a lot of active control over what's going on in the brain. So
Mm-hmm.
ultimately, it's a layer of tissues that houses immune cells, and it's anatomically organized, and the signals that are there can be communicated with the brain. And this is kind of this emerging area, I would say.
Yeah, and I would imagine that we're probably learning more, and I know that some of the things we're learning here about how the central nervous system communicates with the periphery in the periphery. So, we've got cranial nerves and stuff that reach down into the gut, into our organ systems all over the body. And I know that there's crosstalk in both directions there to some extent. The brain can send signals to the periphery to modulate things like immune system function in the periphery. And I would imagine it goes in the other direction as well. Do we know anything about how, if you get an infection or an injury or whatever, there's some inflammation, can the immune system sort of relay that information from like a cranial nerve in the periphery all the way up to the brain and, instigate some kind of preparatory response?
Yeah, so probably one of my personal favorite papers of the last several years comes from Oscar Rolls' lab, where basically what they showed is that if they induce, so beyond cranial nerves, this is direct encoding of peripheral signals in an organized way in the brain. And what they showed is that if you induce a peripheral inflammatory response in the intestines, so gut inflammation, something like that, you can turn on subsets of neurons in the insular cortex, And when that immune response is out of the body, it's cleared, the mice are healthy again. If you reactivate let's say it's like a FOSS trap style approach. If you reactivate those neurons, you actually recapitulate the peripheral inflammatory response. And
Hmm.
it's not a non-specific response. It recapitulates actually the colitis response.
Hmm.
And so in this way, I think that was probably one of the earliest of the recent papers that showing this type of phenomenon that says that there is some sort of organization between how central circuits can modulate peripheral immunity,
Hmm.
and also how the circuits that are the afferent circuits that are going to the brain are encoding this at least in some sort of way that we're familiar with sensory signals being encoded. So it's like this interoceptive code. So I think that's kind of this interesting phenomenon that's emerging.
Yeah, so that starts to get, so it's not just that the brain can send out signals to the body generally that say like be careful, something bad's happening. Particular parts of the body, like specific parts of the gut, the brain can essentially represent a particular type of inflammatory signal and then remember it essentially.
Exactly, exactly. Yeah, so that's kind of this interesting emerging area, and that the way I tend to view this is there's probably multiple ways that the brain is encoding interoceptive signals or like signals from the body. So on the one hand, you have something like that the colitis model, which is more of a wiring diagram. But the way we view the meningeal aspects of signaling is that it's more of this diffusible cue. And so in that sense, it's not actually quite specific for any one circuit unless that circuit has some sort of dysregulation of blood-brain barrier integrity, or if there's a direct link between the released factor and then the receptor expressed in the brain. And so there's reason to think there might be actual multiple encoding strategies that the brain adopts to interpret peripheral cues.
Hmm. Hmm. And then can you talk a little bit about the key immune cells and sort of the overall basic structure and logic of the immune system that is native to the central nervous system, the cells that live in the brain normally? What kinds of immune activity, are going on in the brain?
Yeah, absolutely. So within the brain, we have kind of a few major types actually we're learning of immune cells. I think predominantly the classic immune cell of the brain is microglia. So they're originally derived from macrophages. They seed the brain early in development, and then they're long-lived kind of surveyors of what's going on in the brain. We know a lot about what they're doing in homeostasis and also in certain diseases like Alzheimer's, where they need to effectively clear plaques, things like that. There's also subtypes of macrophages in the brain. These are more specialized macrophages, so things that line the vasculature. They're called perivascular macrophages. And then there's other types of immune cells that I think would be more relevant in certain areas that lack a BBB, such as in the choroid plexus. And so, it really depends on location, but microglia are sort of this, I would say, brain resident type of immune cell that are essentially distributed throughout the brain.
Mm-hmm. And so, under normal, healthy conditions, so we talked about how there's a blood-brain barrier. It's largely preventing stuff that shouldn't get in there from getting in there, but it can be compromised. It can be leaky. And then you just mentioned there's these other parts of the brain and central nervous system where you don't really have so much of a blood-brain barrier, and things can get in. In fact, the hypothalamus can sample the blood because there is kind of a direct connection there at certain points. And so there's some opportunity sometimes for things to get in and out that you might not want to get in. And so we have these resident immune cells, but let's just assume everything's functioning well. It's a healthy individual. You don't have any pathogens from the periphery getting into the brain that aren't supposed to be there. What are these microglia things doing? Are there bugs that are just sort of there in the background, or are they doing other things besides looking for and clearing out pathogens?
Yeah, exactly. So it's largely thought that what they're doing, I mean, so they play key roles in the remodeling of neural circuits. And so it's kind of been classically shown that what they're doing is they can eat or phagocytose exuberant synapses. And when this process is dysregulated, it can lead to different diseases. There's there's this, microglia also play key roles in the metabolic regulation of the brain and sort of what's going on in within circuits. And then, I should add that there's also other types of glial cells that are not immune cells, such as astrocytes, which are sort of CNS resident neuroectodermal lineage cells that are doing similar functions, and these are not immune cells. So they also have the added benefit of sensing the vasculature and also contacting neuronal synapses. And so it's kind of thought that microglia as the resident immune cell and astrocytes as one of the types of glial cells, including like oligodendrocytes and these other types of cells, are involved in just sort of maintaining the environment, actively maintaining the environment of the brain so that neurotransmission can happen properly. So that's kind of how we broadly view the functions of microglia, perivascular macrophages, and other types of glia.
Mm-hmm. So maybe we can start to think about these cells not purely in terms of they're monitoring the environment external to our cells for pathogens like bacteria, but they're just monitoring the environment generally. And even if there are no pathogens around, they might monitor the metabolites that are leaking out of a synapse and all of that stuff, and just sort of sensing what's going on in order to function in some way to keep things, within homeostatic ranges that we want to keep them at.
Yeah, I think that's exactly right. And then I think the way we sort of view it in the context of disease is when you start having perhaps impaired barriers or increased signals that are normally not sensed by the brain or not sensed in high quantities. It's when you start to throw off balance this homeostatic regulation of brain circuit communication as well as the glial regulation of those processes. And so that's kind of how we view the whole system working together.
Mm-hmm, mm-hmm. And so, yeah. And so microglia, these resident immune cells of the brain, they are actually involved in things like the structure of synapses. They can participate in that process.
hmm Yeah, exactly. So so there's a whole cascade that's been worked out that it's thought that there's a deposition of molecules that actually facilitates microglial pruning of synapses that may not be heavily used, let's say. And so that's kind of how we view like what they're doing natively in the brain, at least during development, and in some cases in the context of disease. And so microglia can also release inflammatory factors that can then within the brain start to signal to things like neurons and astrocytes to also modify their functions, which again you would view as linked with metabolism, neurotransmission, and ultimately behavior when you start modifying the tissue microenvironment.
Mm-hmm, mm-hmm. And so when we start to hear things about like neuroinflammation, inflammation that's happening within the brain in the central nervous system, what does that start to look like? How does inflammation manifest within the brain?
Yeah, so I think the way I interpret neuroinflammation, my background is in multiple sclerosis, which is an autoimmune neurodegenerative disease. And so when I think of neurodegeneration or neuroinflammation, that is a dramatic case where the BBB is dysfunctional. It's very permeable to the extent that infiltrating immune cells can get into the brain and also the spinal cord. And not only that, but those immune cells are actively targeting central nervous system antigens such as myelin or aquaporin 4 or other antigens. And so when I think about neuroinflammation, that's how I think of it, which is immune cells definitively infiltrating the brain, where they're releasing cytokines, they're inducing an actual immune response. So there's an antigen that they detect, and they, to some extent, cause neurodegeneration. I guess that's one extreme end of the spectrum. I think there's a much less dramatic example, right? Which would be you kind of have these leakier barriers where perhaps peripheral products or peripheral cytokines can get in. Perhaps they activate microglia or they activate astrocytes. So these are cytokines that are not present normally in high quantities. And then this starts to transform the brain microenvironment and sort of shifts, I guess you could call it the inflammatory set point of a particular tissue.
Hmm.
So there's kind of this continuum, but in my head, neuroinflammation is, I guess a dramatic example where immune cells are getting in, they're releasing tons of cytokines, and I think there's a more of a homeostatic regulation where, there's also low levels of cytokines that can influence circuit function. Yeah, in a more of a modulatory way.
Yeah, yeah. So at one extreme, you've got something like multiple sclerosis. You've got a disease state where immune cells from the periphery can get in, probably in sizable numbers, to the brain, and they're not, or the spinal cord, and they're not supposed to get in there. They don't ordinarily go in. So they're going in, they're causing excess inflammation because they're there and they're not supposed to be there. There's milder cases where, maybe you don't have, an army of immune cells that isn't supposed to get into the brain getting into the brain, but the barrier is a little bit leaky or it's a little bit permeable to a cytokine or something like that. And if, say, someone's chronically inflamed and these things are circulating throughout their bloodstream, or they have an infection and just some of it that's circulating gets into the brain, that can cause some inflammation. And so there's the sort of gradient of inflammation that can come from the periphery into the brain based on the nature of what's wrong, how leaky that barrier is, et cetera.
Yeah, I think that's exactly right. And, but what we have to tease out is, there's in this, along this continuum, there are kind of tunable levels of inflammation that would be not so dramatic as to cause neuron death or something like that, but would be elevated enough to influence the activity of a circuit, which is important for something like mood disorders.
Mm-hmm,
And
mm-hmm.
so that's kind of really what we're interested in. Um, and I could also say, just as a little bit of background, part of the reason we're even interested in that concept in mood disorders is because just reading the literature, patients with depression actually do exhibit elevated inflammatory responses to some
Hmm.
extent.
Hmm.
Again, not multiple sclerosis, but there is kind
Right.
of this low-grade innate inflammation that is detectable. It's detected across meta-analyses of patients. And in particular, it seems to characterize the patients that are not responsive to conventional therapies that are
Hmm.
targeting neurotransmission. And so that's kind of what really motivates us to get after this problem, which is that can we harness immunotherapy targeting the immune system to
Yeah.
modulate circuits and in turn modify mood or affective behavior?
Yeah. I think what you're sort of getting at here is, so obviously we've got things like multiple sclerosis, which are, they're explicitly recognizable as diseases. Like if someone has full blown MS, they're going to have motor symptoms that we recognize as symptoms of disease. We're going to call a neurologist. We're going to get them checked out. You can do the right scans and see lesions that are quite sizable in their CNS. But what you're saying is you can have some level of lower grade neuroinflammation in the brain, and it's not causing massive lesions that'll come up in a scan. It's not causing people to start losing motor control so that we diagnose them with MS or something. But maybe it is modulating circuits that are involved in emotion regulation, and it's giving them low grade or even higher than low grade depression or some kind of psychopathology like that. And we don't normally think of that as being caused by inflammation or the immune system. And we might not even think of it as a disease, or it might be so subtle that we just think someone's in a bad mood or something. But circuit function without cell death and massive lesions can be caused by neuroinflammation.
Yeah, that's exactly right. And so there's actually a whole area of neuroimmunology studying this concept called it's kind of termed sickness behavior, which is
Hmm.
in response to, let's say, a pathogen challenge or something like that, where there's this trigger of systemic inflammation. And again, this is kind of in the absence of a brain disease, right? There are these behaviors that change in individuals across, the evolutionary the span of evolution, where actually it changes motivation, it changes homeostatic behaviors, it changes reward behaviors. And, there's been some great work lately studying this, I would say, from Catherine Dulac's lab, Jeff Friedman's lab, Steve Lieberle's lab, where, you can actually induce this peripheral inflammatory response and see dramatic changes in the activity of circuits underlying homeostatic behavior or just limbic areas that influence mood and motivation. Um, and so there are these like intimate connections that we know of between the immune system and the brain when the immune system is engaged. And so if you think about pathology, when perhaps the immune system has been activated or aberrantly activated in the past, and some of these peripheral responses are engaged continuously, if you have these molecules that are being produced, they could influence circuits in a similar way.
Yeah, yeah. And then, obviously we know that chronic inflammation is basically been a growing problem for a long time. So a lot of people that don't have overt pathologies or a disease that can be diagnosed as a discrete, pathology, low grade inflammation, medium grade inflammation, sort of background inflammation is experienced by a lot of people for various reasons that we could spend, whole podcasts talking about. But another area where there seems to be some convergence here that I think is going to tie into some of this new work you've done has to do with psychedelics. And in my read of the literature, one aspect that I think is really interesting about some of these psychedelics that's underappreciated is that some of them are actually quite potent anti-inflammatories. We know about their psychedelic effects and the hallucinations. We know about how they're being used to treat certain psychopathologies like depression. But I've had people like Charles Nichols on the podcast, and he's done really interesting work showing that actually they can act as really potent anti-inflammatory compounds in some cases.
Yeah.
So what is the tie-in here, and how does that set up some of the recent work you've done?
Yeah, exactly. So yeah, I agree. Charles Nichols' work is some of the work that inspired us once we sort of made some of these early observations. And the idea that we have here is that the way we view psychedelics and inflammation, it really emerged from, as we did more just experimental work, but it's kind of like Gould Dolan's work showing that there's kind of this critical period where they're effective. And so that's how we view their activities in regulating inflammation. So, in other words, some of the work we found is that on certain genes, psychedelic in immune cells, let's say, psychedelics don't really have an effect. So there's really not direct modulation. Yet, if you co-administer other compounds, like in our case, we did corticosterone, the stress hormone, or we did inflammatory cytokines, things like that, then there's this critical window of signaling that's engaged. And within that window, psychedelics are effective. And in some cases, if you go beyond that, so if you dial the system too high or too much inflammation or too little inflammation, let's say, psychedelics don't have this kind of anti-inflammatory property. That's kind of like how we
Hmm.
interpret it.
Hmm.
Yet, if you use them in the right window where either a tissue is dysregulated or cell signaling is dysregulated, that is when they actually exert beneficial effects. And so we're really interested in that concept because we think it lies within intracellular signaling and how coincident signals prime cells to be in a certain state where engagement of these pathways activated by psychedelic would be effective. So I think broadly that's sort of how we view it, if that sort of explanation makes sense.
Yeah, yeah, I think it does. And so if I tie some things together here for people, so Gould Dolan's work's very interesting. I've had her on the podcast, and so I'll sort of articulate how I think she would describe her work, and basically how you just did. And there's sort of there's some controversy in the field right now, and some different ways of thinking about this, but one way that some people think about psychedelics is that, okay, we see these neuroplastic changes, we see these potentially therapeutic changes. One way that some people think about that is the psychedelics themselves are inducing the plasticity per se and having all of these effects. Gould Dolan's work starts to say, well, actually, it's not quite like that. These psychedelics are opening, they're more of a permissive signal. They are creating metaplasticity, so they're enabling plasticity to happen if it's stimulated in a critical window around the time the psychedelics are administered to induce the plasticity and the circuit change and things like that. So it's this somewhat subtle difference in viewpoints. And what you're starting to say sounds like it's kind of similar to what Gould Dolan has seen, whereby you can induce a sort of metaplastic shift with something like a psychedelic, and then another compound, another environmental factor can impinge on the nervous system, and it's rendered more effective at driving change when it's used in conjunction with the psychedelic window that opens.
Yeah, I think that's sort of how we view it. I mean, the way I was trying to state it from the perspective of how the compounds would be acting on immune cells or immune cell signaling, or even like tissue physiology more broadly. But that is kind of um, how we sort of view it, which is there are these kind of coincident signals, at
Mhm.
least in something like primary immune cells or tissue responses or some aspects of behavior. But there's obviously no doubt that there is also this induction of things like new spines or something like that caused by psychedelics. But it seems that when we use them in in immune cells, or at least to target immune tissues, it's the tissue already has to be at a certain state. And sort of what I meant by that is when we would treat animals with these compounds, the animals had to already have dysregulated, let's say, meningeal immune responses in
Mhm.
order for psychedelics to reduce the abundance of immune cells in the meninges. And it also appears to be specific to certain tissue states. So in the context of stress, what we saw is, if you administered psychedelics to animals that underwent chronic stress, but then looked at their spleen, there really wasn't so much different in response to psychedelic treatment. So it seems to be, at least on the tissue level, is kind of how we're thinking about it. There seems to be this tissue dysregulation that needs to have happened. And I assume there's an upper and a lower bound. That's why I call it a window in my head, for these compounds to be effective at reducing it. But of course, we think that they are also per se reducing some aspects of inflammatory signaling in the cells themselves as well.
Mhm. So for some of the things we'll talk about, it sounds like this isn't just sort of a broad spectrum, hammer. It's not like the psychedelics are just turning down inflammation everywhere in all tissues. There's probably a lot of context and tissue specificity as to whether or not you see some of these effects.
That's a much more succinct way of saying it. Yeah, exactly.
And so, set up this paper for us a little bit. And let's start with the term psychedelic itself, because that's also a term that people have different views on. When
Yeah.
you used what psychedelics did you use? How are you using that term, and what are the specific drugs we're actually talking about here?
Yeah, so we used the term psychedelic because really we used it for simplicity, and we sort of state that in the paper. We appreciate that there's kind of different classifications depending on, the pharmacology or the structure of the compound and what receptors you're hitting. But really we considered we use psilocybin, which is the component in magic mushrooms. It's kind of what's considered a classical psychedelic, because it's engaging the type 2A serotonin receptor. And then we also used MDMA, which is structurally distinct and also has different mechanisms of action. It's thought to, at least partially also be modulating serotonin signaling, but by a different mechanism. And, depending on how you can call it, you could call it an entactogen, empathogen, things like that. But I can tell you just the reason why we chose the two of those. We
Mhm.
chose them because, first of all, they've both been used in clinical trials of patients with affective disorders. So, psilocybin has been used in depression, and MDMA has been used for PTSD. And so we thought that these would be clinically relevant, therapeutically tractable compounds to work with.
Yeah, yeah.
And yeah,
Yeah, so it's two different substances. Psilocybin from magic mushrooms, that's yeah, the classic hallucinogenic psychedelic compound from mushrooms. MDMA, certainly strongly psychoactive, but most people don't really call it hallucinogenic
yeah,
or psychedelic in the so-called classic sense. But then again, if we bring up Gould Dolan's work, part of what she showed is despite the fact that these are different drugs with different effects that act through different mechanisms, they actually have some convergent mechanisms downstream that tie into this metaplasticity idea. And
exactly.
so I think from that standpoint, you would expect them to have similar effects at some level. And okay, so you used those two drugs. What exactly did you do for the main experiments here? Can you describe them in a basic way?
Yeah, so again, I think tying this all back to the kind of the neuroimmune interactions mechanism. What we really did was we subjected mice to a chronic psychological stress paradigm, where we did something called chronic restraint. So we restrained them in a small space, for longitudinally. So we did this for basically 1 to 3 weeks, and we were looking for a time point where the mice showed dysregulated behavior. We focused on fear, as well as dysregulated immune responses, and so we settled on this paradigm that's approximately 3 weeks in duration, where the mice are taken in and out of the tube every day. They spend about 6 hours a day in this tube, and they get stressed out. And this was the time point where we detected these kind of convergent immune and behavioral changes. So that's sort of what we focused on.
So what are the behavioral changes? Are they no longer, are they extra fearful? Are they not responding in the normal fearful way?
Exactly. So the mice essentially are extra fearful. So if you do a classical fear conditioning and then you look at how the mice respond in essentially a new context, the mice exhibit greater fear, let's say generalization, if you wish. And we validated this using, kind of two independent paradigms. And so from there, we were hypothesizing that perhaps the fear center could be, which is the amygdala, could be one of the dysregulated circuits.
Mm-hmm, mm-hmm. And then how is the immune system dysregulated here? What does that look like in these animals?
Yeah, so initially we just did something very simple, which is we surveyed cytokines in the plasma, and it's really just looking for kind of changes in these molecules, which other labs have shown occurred in response to chronic stress. I mean, or even acute stress. So this is work by Scott Russo or Phil Swirsky, a lot of people, showing that there's kind of these massive changes in cytokine production or leukocyte shifts. But also there's changes in tissues across the body that produce these cytokines. And so this was sort of just a first approximation, but we wanted to get at how those signals might be actually influencing the brain. And so that's kind of how we got to the time point where we saw immune changes. But then we looked we basically just surveyed the abundance of different immune cells across different tissues in these stressed mice, trying to figure out which specific subsets would be changed in different compartments.
Mm-hmm. What were some of the largest particular changes that you saw?
Yeah, so what was interesting is we actually saw within the meninges, there was recruitment of multiple different classes of immune cells. One of the populations that we focused on is something called a pro-inflammatory monocyte. And these are cells that are pretty short-lived, but they are very important in modulating tissue inflammation. They also kind of come in different flavors, but this particular type of cell is known to traffic to tissues. It can either circulate out, it could die, or it can also differentiate into a more long-lived type of macrophage. And so they're in circulation until they sort of encounter a tissue, which could be something like the meninges.
Hmm. I see. So they're induced by some stimulus. Depending on where they are and exactly what's happening, they might sort of be around for a little while and then go away, or they can change into a different cell and stick around for extended periods of time.
Yeah, exactly. And these cells are thought to be liberated from the bone marrow in response to, or other tissues in response to chronic stress. And it kind of really depends on how long the stressor is in terms of where these cells go, what they do, and so on.
And so are monocytes, are they like white blood cells that would eat pathogens, or like what do they do functionally?
Yeah, exactly. They're kind of recruited to sites of wound healing, if you wish.
Mm-hmm.
And there, the kind of the tissue instructs them on what to differentiate into, where they can kind of take on more of a reparative role. If there's a pathogen or something like that, they can also secrete kind of these innate cytokines, or also, and also differentiate into, let's say, like types of macro tissue resident macrophages or things like that.
I see. So they're
So
almost like
yeah,
they've almost got kind of like a stem cell vibe where they can become different things depending on the conditions. They might clean up debris, they might deal with
exactly,
pathogens, whatever.
exactly. Yeah,
Hmm.
that's right.
So how do we get into the brain here?
Yeah, right. So one of the things we began doing is we were looking for basically once we settled on the amygdala, we were starting to look for changes in the brain associated with these time points where we saw kind of, let's say, convergent neuroimmune interactions. And so one of the cell types we focused on are astrocytes, which I mentioned earlier, these resident glial cells of the central nervous system. They interact with both the vasculature, so they sort of have these end feet that wrap blood vessels, and they also directly interact with synapses, so they can kind of actively shape neurotransmission. And so, through a series of experiments, what we found is that there's a receptor in astrocytes, a receptor tyrosine kinase called EGFR, which actually appears to be sensitive to the same cytokines that we detected in the plasma. So basically, if you are looking at how EGFR expression changes or molecules that EGFR controls, if you treat with steroid hormones and IL-1 beta, let's say, from which is a cytokine we detected in the plasma, it actually modulates EGFR expression, which in turn then shapes other transcriptional programs that we investigated. So basically, we view this receptor as kind of, first of all, sensitive to inflammatory cytokines, but then also what we know from many decades of work studying this receptor is that it is kind of a buffer against inflammatory signals. And so
Hmm.
when it's down-regulated, you can actually intracellular signaling that's inflammatory, driven by something like NF-kappa B, can start to take root and then engage the expression of molecules that are not typically highly expressed.
I see. So EGFR, it's a protein that can help buffer against inflammation. So it can help prevent cells from getting messed up from inflammation. But if it goes down, that means they're more sensitive to inflammation.
Exactly, exactly.
And so, what did you see in terms of how the use of psychedelics sort of tied into this? Like, I can maybe anticipate the answer just given what other literature has seen. But why don't
Yeah.
you tell us what happened and how it ties into this EGFR thing?
Yeah, exactly. So what we did really is we examined how, first of all, the immune system modulates this signaling, this astrocyte circuit.
Yeah.
And so basically we did a series of experiments. I think we did four different experiments, which all kind of said the same thing, which is if you give mice more monocytes, more inflammatory monocytes, so if we transfer them into mice and every day and then stress those mice out,
Hmm.
we actually see first of all, recruitment of these monocytes to the meninges, and we see elevated inflammatory responses. And we also see downregulation of EGFR signaling in astrocytes within the amygdala. Conversely, if we deplete those monocytes, which we did chronically using a genetic strategy, so the monocytes have just been deleted for the entire duration of stress, or if we acutely ablate them specifically within the meninges using a monoclonal antibody, you can also tune EGFR signaling in the amygdala. So if you get rid of monocytes, you can actually boost EGFR activity. So it seems to be responsive to peripheral inflammation, and it in turn sort of
Mhm.
is influencing fear behavior.
Yeah.
So the psychedelic part comes in because what we find is that in the stressed mice, where they have this accumulation of immune cells in the meninges, when we administer psychedelics, the same immune cells that are modulating EGFR leave the meninges after about a day.
Hmm.
And so, so what we were thinking is that psychedelics are actually these not anti-inflammatory, but let's say modulators of tissue plasticity by essentially getting rid of cells that can control, basically getting rid of immune cells that can control behavior.
Hmm.
So that's sort of how we view it. It's a complex multicellular circuit, but that's sort of how we view them as sort of indirect modulators of what's going on in the brain by influencing the immune system.
Yeah. So restraint stress can increase the amount of these monocytes that are circulating. You guys can also just add monocytes into the system in different ways. But when these monocytes go up, the normal stimulus for that is stress. If they get to the meninges, they can have this effect on the brain via EGFR, where they're essentially rendering circuits more vulnerable to this inflammation induced by stress. But if you get rid of them in the meninges, if you prevent them from getting there, or you use psychedelics, they basically leave there earlier, and then the brain is not rendered sensitive to inflammation so much.
That's right. And I think more broadly, the whole monocyte inflammation concept ties into other work also on the amygdala done by Jun He and Gloria Choi, where they showed actually there's this bidirectional modulation of anxiety or amygdala activity, if you wish, that is controlled by pro and anti-inflammatory cytokines. So the
Hmm.
pro-inflammatory cytokines, something that would be released by monocytes, et cetera, drives this fear behavior. And then if you administer anti-inflammatory cytokines, or if you get rid of anti-inflammatory cytokines, you can then modulate in the opposite way fear behavior. So it's really this push and pull, the kind of way the way we would classically view pro and anti-inflammatory cytokines operating in the immune system as also operating on different types of brain circuits. So I think that's kind of this interesting interplay.
What so the immune system can have this kind of this effect on the nervous system. Stress can, it can trigger the immune system to react in some of these ways. So, chronic stress is a thing a lot of people experience. Chronic inflammation is something that a lot of people experience. Is sort of the story or the picture here that's emerging more or less that if you're experiencing chronic stress of some kind, if you're experiencing chronic inflammation of some kind, whether that's coming from an environmental stressor, something in the diet, all of the above, any number of things we could point to, this can actually have effects on nervous system function and things like mood that we might not ordinarily associate with immune system stress.
Yeah, I think that's exactly right. I mean, it ties into this concept that there's a kind of unanticipated brain-body communication. So what's happening in the environment can profoundly shape what's going on in the brain. Um, and we know these types of changes happen. So patients that are receiving certain types of therapy for different diseases like immunotherapies, it directly modifies their mood. That's just kind of a more extreme example. And so there's it's unclear, I would say in humans, how much of these environmental challenges are necessary to evoke the same sort of circuit changes that we see in the animals, how chronic they have to be, how intense they have to be, whether it's even the same molecules that are the same risk factors. But I think there's kind of an underlying principle, which is exactly what you hit on, that what's going on in the environment, if it's shaping processes in the body, eventually those signals through the conduits we talked about earlier, like the meninges or even the vasculature, those signals can kind of get back to the brain and start shaping how behavior is modulated in kind of this unanticipated way.
Hmm. And yeah, I mean, it's easy to think about how some dangerous loops could get formed here. If you've got some chronic inflammation that's making an animal more fearful, that might make them more prone to stress and just detecting fear in their environment, which is going to keep them in that more inflamed state. And it's not difficult to imagine how an animal, human or otherwise, could get trapped in this sort of vicious cycle.
Yeah, I think that's right. And so the question then becomes sort of therapeutically, what can we do or when can we intervene? And I think for us, one of the things that we're excited about is if there's a way to prevent some of these cells from trafficking to the brain in the first place, can we sort of and of course we would need to identify the right patients that this would be effective. Can we sort of abrogate some of these changes? And then, getting at the larger picture, what are the circuits that we would be targeting with immunotherapy and mental health? Is it non-specific circuit control, or is it kind of different populations of cells that are communicating with different types of circuits?
Mhm.
That's kind of, I
Mhm.
think, the next big question.
Yeah. So remind me, what the main effect of MDMA and psilocybin here was basically to cause the monocytes to leave the meninges earlier, and therefore they would have less of effect on the fear circuitry in the brain.
That's right. Yeah. And so the way we envision this working, the question was kind of, is this a direct effect? So is it
Right.
altering their chemotaxis? Or is it an indirect effect? Does it involve other cell types? And we sort of tested both possibilities to some extent. So we do see that, yes, if you take the cells out of the animal and treat them with these drugs, they respond to psychedelics directly. In vivo, it appears to be a more complicated picture. So the way we actually think it might be working is what we detected is there's kind of this increased vascularity in stress, which we measured by just looking at the animal's head using a vasculature binding dye.
Hmm.
And we see kind of this stress-induced vascularity, if you wish. But we could block that by treating the stressed mice acutely with psychedelics. And so the way we envision this happening is through a vasoconstrictive mechanism. So basically, stress induces vascular dysfunction or vasodilation, something like that, where you simply have more immune cells that are able to be in or around the vasculature of the brain. But then psychedelics are known to cause vasoconstriction, and
Yeah.
then it decreases the space that these cells can even occupy. And the last thing I just want to say is we tested that as well because we gave mice psychedelics together with a vasodilator. And when we administered the vasodilator with psychedelics, we actually reversed this psychedelic-induced decrease. We actually just
Mm.
saw more monocytes in the meninges. And so that's how we sort of think this might be working through kind of both potentially direct, but also probably also major indirect effects.
Interesting. So psychedelics have this effect. They can make the monocytes go out of the meninges, but you see these vascular changes. And if you induce vasodilation simultaneously with administering psychedelics, this effect goes away. So it suggests that it's through some vascular effect that the psychedelics are working here. And remind me, so I know that psychedelics have these vascular effects. Do those happen through the serotonin 2A receptor, or do they happen through some other mechanism?
Yeah, so that's I think where it gets complicated. So it's at least partially involving the sympathetic nervous system, which densely innervates the meninges. And I actually don't know all the literature on how, what specific receptors are required for the vasoconstriction, whether it's strictly 2A dependent. But what we think is that perhaps at least sympathetic neurons are modulating this feature of the meninges or even the brain vasculature. Um, uh, by when they also respond to psychedelics and stress and things like that. And so because when we got rid of sympathetic neurons, we this was a complicated experiment because it took about a week to get rid of the neurons. But the point is that psilocybin seemed to have no more effect on the meningeal egress of immune cells. So it would suggest that psilocybin's effects are at least partially dependent on sympathetic neurons, but MDMA still caused this egress of immune
Hmm.
cells. And again, we looked at sort of immune cells more broadly because it took a week to even do this, and monocytes survive for about 3 days, something like that.
Yeah.
And so there seem to be compound specific effects when you look on the sympathetic side. So there's probably this interplay between sympathetic nervous system, cell autonomous vasoconstriction or not, and also immune cell modulation.
Yeah. So they're having the same sort of MDMA and psilocybin are having the same overall effect on monocytes, causing them to egress here. But it sounds like the details probably differ, and that still needs to be worked out. So on the one hand, the same sort of overall effect is seen, but on the other hand, they're not working in identical ways.
Exactly. I think that's exactly right. So we're really interested in, I guess, two things, like looking at, on the one hand, structurally similar molecules to each compound and seeing the relationships between what we found and what each of these other molecules do. But then also looking in other tissues, because I think what's very exciting to us is, these compounds are great in psychiatry, but when you have a neuron or an immune cell or a structural cell, something like glia or just stromal cells, together in a unit, that's a feature that's present in tissues throughout the body. And so we think that there might be contexts where these compounds would have great utility to cure aspects of tissue pathology in diseases that we weren't even thinking about because of the mechanisms are generalizable across tissues.
Hmm. So what are you guys working on to follow this up? What are some of the next steps here?
Yeah, so we're interested in a few things. So the one of the things that we're doing is we really want to understand in the context of, let's say, psychedelic induced plasticity, to what extent in stress, let's say, to what extent is the immune cell component modulating that plasticity? It's a complicated question because we didn't really get at that specifically. We sort of looked at it indirectly. And so we really want to identify if, the time scale or the strength of how immunomodulation would influence the rescue of behavior. Alex Kwan just had a paper come out, I think a few weeks ago in Nature, just, beautiful paper showing how dependent the process is on, I believe, the PFC neurons. And so, but then, how does the immune system fit into that type of thing? I think that's something we really want to work out. The second thing we're doing is related to what I said earlier, which is we really want to understand to what extent these sort of cells and the principles that we find here are applicable to other diseases or other tissues. And so we really are interested in models of inflammation and how these compounds could be useful. And that extends also to looking at how patients or how it modifies inflammation in patients.
Yeah, yeah. And then, at the beginning, you mentioned that, there seem to be certain forms of depression, potentially even the ones that don't respond as well to medications that are tied to neuroinflammation of some kind. What do we know about neuroinflammation and sort of immune system brain crosstalk in human psychopathologies that have to do with stress and depression?
Yeah, so it is complex because depression is so heterogeneous.
Hmm.
But what we do know, for example, if you look at biomarker studies, there have been meta-analyses that showed elevation of plasma cytokines that are enriched in depressed patients relative to controls across multiple studies. To some extent, the changes in these profiles are associated with therapy outcomes. That seems to be also the case. And then there appears to also be, at least looking at some patient-based in situ studies, there seems to be some evidence that there's at least local low-grade innate inflammation in certain brain areas that are important in depression and also in affect in mood, that needs to be followed up on more, I think. So I think we're just kind of scratching the surface at this point, and we really need to dig in to specific mechanisms and stratifying how one mechanism might be good for one patient subset, but not another. So I think we're really just in the early stages.
Hmm. Hmm. And, what do you think about so you did this work with psilocybin, MDMA. On the one hand, they have very different mechanisms, but as we discussed, there's some reason to think that they could have similar effects despite those differences in mechanism. That ties into Gould Dolan's work and how some of these things, despite interacting with different receptors, can sort of converge downstream in different areas to affect things like plasticity or the ability to induce plasticity. Are you guys looking at other drugs that are, broadly speaking, considered psychedelics or psychedelic like? Or do you think these two are worth focusing on for some reason?
Yeah, exactly. So we are absolutely doing that. So we're interested in other structurally related molecules, some of which aren't even Schedule 1 or psychedelics,
Hmm.
that are related to, let's say, psilocybin.
Yeah.
Um, and we're also interested in particular in other psychedelics or psychedelic-like compounds that are clinically relevant. An example here is something like ibogaine,
Hmm.
which, if I'm not misstating things, has been shown to be effective for eating disorders and also brain injuries, which carry a lot of brain inflammation.
Hmm.
And so we're interested in these compounds because of kind of these phenotypic properties that they might be relevant in people, and we want to understand a little bit better what they do in animals.
Interesting. And so, for the effect that you saw, where you have these monocytes getting into the meninges, where they can then induce these changes in the brain, but then MDMA and psilocybin can sort of interfere with that and get them out of the meninges. Does that imply that uh, so for MDMA and psilocybin to have that effect on the monocytes in the meninges, that does not require them to cross the blood-brain barrier and go into the brain. Is that right?
Exactly. So this part appears to be where we're thinking of let's say neuroinflammation in a stress context to be distinct from something that would happen in neurodegeneration. And so the barrier is just weak enough where peptides can get through, something like small peptides like cytokines, but it doesn't appear that the cells themselves get through. So there's evidence, for example, in the nucleus accumbens, where there's also this BBB breakdown, where the immune cells are in the vasculature, the monocytes are in the vasculature, but they don't appear to be actually getting in. And that's basically what we saw. We didn't really detect them inside of the brain, but they were able to modulate what was going on from essentially the other side of the barrier. But just to say, I mean, some of this could be a detection issue,
Mm-hmm.
because, like I mentioned before, these cells can differentiate into tissue resident cells. So it could be something like that happening, although we didn't detect many more macrophage-like cells in the amygdala, but that's not to say it can't happen. For example, in some stress paradigms, T cells have been observed to be in the amygdala. And so we really just need to understand, how this is all working together, and what in what context can the cells get into the brain versus signal from outside.
Mm-hmm. Mm-hmm. And when we talk about things like blood-brain barrier integrity and neuroinflammation, does the blood-brain barrier just tend to naturally sort of decline in its integrity with age? Do people tend to get leakier over time? And if so, like, are there clear major drivers of that, or is it something that just happens for, 1,000 different reasons?
Yeah, exactly. I think so aging is this, big mystery. So I think it does happen over time. But I think it's multifactorial. And kind of to what we were discussing earlier, I think there's kind of differential susceptibility of some individuals to, certain environmental factors versus others, depending on sort of where the BBB might be more open. But this sort of leakiness characteristic does appear to be kind of like a feature of aging, and it's associated with dysregulated immune responses, aging.
Mm-hmm.
So I think this is a key feature that we have to think about in terms of susceptibility to these types of diseases or changes in behavior.
Yeah, and so I guess one of the last things I'll just ask you is, so are you pretty excited about psychedelics broadly speaking, to include things like MDMA? Do you think the excitement around these things is warranted, or do you think people are getting a little exuberant? What is sort of your take on whether or not we should be, going in the direction people want to go from perspective of looking for FDA approval and taking these things to the next level?
Yeah, I mean, so I think my perspective is, what excites me first and foremost is the clinical data,
Hmm.
which I understand has its caveats because of placebo controls and so on. But that's what excites me first and foremost, and so that kind of guides my excitement. So yes, I'm broadly excited about what they can do. Um, I think the fact that they act on multiple cell types is also very exciting, and to control sort of what I view as more like tissue physiology,
Yeah.
in addition to plasticity. On the other hand, I don't view them as a cure-all. So, we were talking about these windows. I do think there's contexts where they're very effective. Again, we have to figure out all the rules of this. But I don't think they cure everything. But where I think I get the most excited is to start engineering next generation psychedelics, which is, I think, something people like Brian Roth are doing, which is, you can create these bespoke libraries of compounds, test them, do all the pharmacology, and then engage the same pathways, potentially in a better way, than your natural products. And then really try to engineer kind of therapy, some of which might be better to act on the brain, some of which might be better to act on peripheral tissues. And so that's where I think I get excited, is like, first and foremost, starting from patient data and then trying to reverse engineer the compounds to be better and more effective without
Hmm.
any of the side effects.
Is there anything you want to reiterate for people from what we discussed, or any final thoughts you want to leave them with about this subject, neuroimmune interactions and the like?
Yeah, I think the thing that we find surprising, very exciting, is that the brain and the body are in intimate communication. We think that there are probably a different set of rules governing these interactions. And I think that really identifying the cellular and the molecular logic between communication of the body with the brain and vice versa will, I think, open up kind of like new eras for therapies, because it'll give us access to cell types that modify another organ system in completely unanticipated ways. So I think that's the first thing we're very excited about. And the second is what we just discussed, which is like we don't think that these drugs are potent anti-inflammatory agents like NSAIDs, or they're not going to cure everything, but we think that they're engaging signaling that's broadly relevant for many diseases, and we really just want to understand what they're doing and how they're doing it.
Mhm, mhm. All right. Well, Michael Wheeler, thank you very much for your time. Very interesting stuff.
Thanks so much. I appreciate it.
Visit mindandmatter.substack.com to find all of my content. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle. And you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling. And the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today.